So today, we're going to be talking about metabolism of other carbohydrates, okay? So today's topic is going to be we're finishing carbohydrate metabolism today, and today we're talking about metabolism of other carbohydrates. And this thing called pentose phosphate pathway, okay? You're going to hate the pentose phosphate pathway because, it's really confusing. And again, the reference material that I use are listed down here. One of them is your textbook called Biochemistry IV by Matthews, and these couple other guys. And also, when I was a college student, I used to use this book called Leininger Principle of Biochemistry. If you guys are going to do like biochemistry in the future, I still recommend you get this particular book, Leininger. Leininger is actually a very famous book. So back when I was at college, University of Maryland and UCLA, we used to use this book. The reason why we don't have you guys use this book is because it's slightly more expensive now. And also Biochemistry IV by Matthews does a pretty good job on the chemistry aspect. Okay, so that's one of the reasons why we chose Matthews. Okay, utilization of other carbohydrates. Okay, so this is actually a very interesting slide. Basically, these are some of the different types of carbohydrate sugars that we will get exposed to in our daily diet. Okay, so biochemistry is for you to understand the translation of energy. Okay, so how do you get energy? And basically, starch we talked about. Okay, at the very beginning of our class, we talked about starch. Okay, starch like your bread, that kind of stuff. And starch gets degraded into glucose. Okay, so glucose. So this guy. Okay. This guy can go like that and then go to glucose. Glucose gets consumed by your cells, and then that is going to use hexokinase to then activate into glucose 6-phosphate, and G6P cannot leave the cell. We talked about that. Now, a couple other things that we're going to talk about. One is let's start with maltose. Okay, maltose, 麦芽糖. Okay, do you guys remember back last semester when I was teaching you guys the nomenclature and the general structure of carbohydrates? Did I mention that maltose is not a naturally occurring product? 有没有听懂我意思？就是你没有植物会喷麦芽糖啊，就是应该不太有。Okay, maltose is a dimer of glucose, alpha glucose. Okay, so to my knowledge, I don't think there's like a plant that just spits out maltose. Okay, but how do we get maltose? You get starch and you heat it up. Okay, you heat it up and starch degrade, and it's going to degrade into maltose, right? So maltose, if you eat it, it's very sweet and very viscous. You eat it, we're going to have an enzyme called maltase for degrading maltose. Okay, maltase degrading maltose, right? So you gotta appreciate biochemists for making easy names for you. All right. So maltase degrades maltose, and maltose then becomes 2-glucose, and glucose continue for glucose 6-phosphate. That's fine. Lactose. Lactose is the sugar found in dairy products like milk and cheese. Okay. Or if you really like cream, sometimes they also have a lot of lactose in it. So lactose is also a dimer. Okay, it's disaccharide, and it's a dimer of galactose and glucose. So it's going to use an enzyme called lactase. We'll talk a little bit about lactase. Lactase then hydrolyzes lactose to make glucose, and glucose, it goes. The other sugar it makes is galactose. Okay, galactose. And galactose, the metabolism for galactose is slightly complicated, which we'll talk about in a little bit. So in many, many, many steps, eventually it also converges into glucose 6-phosphate. Okay, pretty much the same thing. Right? Sucrose, my personal best friend, because it tastes sweet. Of course, people don't really use sucrose in your handshake drinks. Okay, 手摇杯应该都是 fructose. But anyways, sucrose, if you drink sucrose, typically it's in a solution, But if you can buy it, it's powder, but usually it's a solution because it's extracted from sugar canes and sugar beets. Sucrose is a dimer of glucose and fructose. Okay, and obviously glucose, you know, it goes this way. Fructose, okay, it's interesting. Fructose has two different mechanisms for degradation, okay? One is very simple. You use hexokinase, okay, directly go to fructose 6-phosphate. Another mechanism is you can generate this fructose 1-phosphate. Okay, we'll talk about the difference between those two, okay? But what you should notice, there's another thing called mannose. I don't know where you get mannose from, I forgot, but mannose is around. What you should notice is that you see hexokinase in both glucose metabolism and mannose metabolism and fructose metabolism, okay? So, you know, this same enzyme can be used for different hexos. First of all, I'd like to talk about lactase and lactose intolerance. How many of you guys are allergic to milk? Anybody allergic to milk, right? There's a population, there's a small portion of the population in the world. Actually, if you look at this chart, not that small, it's actually quite large. So there's a population in the world that does not take lactose well, okay? And the distribution of that population is shown in this map. I should have put the reference here, but you can get the reference. This food intolerance network, okay, whatever. So basically what happens is in the world, and if you look at population by region, and you know the denser the color pretty much means that the more prevalence of lactose intolerance. Okay, for those of you that don't know what lactose intolerance is you drink lactose and your body can't really metabolize that well, so then you like have diarrhea. All right, so diarrhea, this is how it happens. Okay, so basically you need to have an enzyme called lactase, okay, lactase, that can degrade lactose into galactose and glucose, okay? Humans have to have this, otherwise we cannot degrade lactose, and our cells can't just directly utilize lactose, okay? Our cells can't, but we can utilize glucose and galactose, right? So what happens is that if you have lower lactase production inside of your body, okay? So if you don't have lactase in your body, then what happens is you do not degrade lactose, and this lactose will get degraded by microbes that can consume lactose, and then they will produce fermentative products such as lactate, H2, methane, etc. Okay, so that's why you get gassy, okay, because they make a lot of gas. So then you have gassy feeling, and that's why you have to go use the restroom. Let's take a look at this picture. Normally in your small intestine, if you have good amount of lactase, you will be able to degrade lactose into glucose and galactose. That's normal. But if you don't have enough lactase, we call lactase deficient, okay? So you don't have lactase, so instead lactose will get degraded by bacteria, okay? And bacteria will create gas, okay, so gas like H2, CO2, methane, those type of stuff. And also it produces organic acids like lactate, these fermentation products, okay. And lactate, because it's an acid, okay, it's going to draw water, right, because through simple osmosis, it's going to draw water into here. When you have gas, water inside your intestine, okay, you want to take a dump, okay? So some population in the world, then we look at why, like China or pretty much Asia, Africa, South America, these places have higher percentage of lactose intolerance. In addition to any genetic defects, what happens is that most mammals will decrease in production of lactase when entering adulthood, okay? So as you get older and older, this is an evolution thing, okay? When humans are first born, okay, little babies usually eat milk, okay, from like parental, like mother milk. And as you grow older and older, you can start eating normal food. So the intake of lactose naturally decreases, okay? So through evolution, basically the cell is trained to not make so much lactase, right? Because back when, like maybe 2,000 years ago, we don't have that many cows, you don't have that many milk. In those cases, you just stop eating like dairy stuff, Okay, stop eating milk-based stuff. And so through evolution, we don't want to waste energy to produce an enzyme that we don't use. Okay, so we stop producing lactase, okay, as we enter adulthood. Okay, however, it should be noted that this effect is not as pronounced, so not as severe in people with a diet that continues to contain milk and milk-related products. So a lot of European countries, you see how like Europe here and America. Okay, so these people, they eat cheese like every day and they really like, love cheese. So they continue to take lactose, and because of that, they're training their cell to continuously producing lactase. Okay, so then they will not be lactose intolerant. Okay? So lactose gets degraded into glucose and galactose. Okay? Now you know the metabolism for glucose already. Now the other monomer that it spits out is galactose, and galactose gets converted into glucose 6-phosphate. How? First, you should recognize that galactose and glucose, they're really a diastereomer. Okay, remember the definition of diastereomer is the same chemical formula, but you have one chiral center that is different, right? So the different stereoisomers, you have enomer, diastereomer, and epimer. Typically start with this beta galactose. Beta galactose, okay, so this guy is beta. You have to first use a mutarotace, an enzyme mutarotace that can sort of turn beta into alpha. Okay? Why do you need to do that? That is because the kinase, the galactokinase, is specific for alpha galactokinase. Make sense? Right. So then you turn alpha galactokinase, okay, and you activate that. Now you get alpha galacto 1-phosphate. Okay. Notice the 1-phosphate. And then in step 3, you're going to turn this alpha galacto 1-phosphate into glucose 1-phosphate. Okay, it's not magic. Okay, it's actually chemistry. What happens is instead of using epimerase, okay, because basically the only thing that changed is here, right? So instead of using the epimerase that can sort of chirality, okay, instead of using that, you're actually going to use a transferase. And this transferase, you get that, this transferase is going to sort of convert galactose 1-phosphate to UDP galactose. And then what you get is a UDP glucose to a glucose 1-phosphate. Okay, so you swap the marker. Okay, here is a phosphate. At the end, you get glucose 1-phosphate, glucose 1-phosphate. You can then do a mutase, which then generates glucose 6-phosphate. Okay? Now, then you might be wondering, where do you get UDP glucose from? Okay, you can get that from 2 places. One is you take glucose 1-phosphate like this, okay, and you use UTP, and then you pop UMP onto this phosphate. Now you have UDP. Once you have UDP galactose, okay, then you can do the epimerase. And that will change you from UDP galactose to UDP glucose. Now the question then is, why can't you just do it to this guy? This thing is called the Lelor's pathway. It's discovered by this guy. Okay? So galactose metabolism, at least in humans or in animals, it's more complicated. All right, it has to do, you have to first change the stereochemistry, you activate that. After you activate that, you have to change it into UDP. Here is, oh no, yeah, you have to use the UDP thing right here, and then you then change it from galactose 1-phosphate to glucose 1-phosphate to glucose 6-phosphate. Okay? So it's a little complicated. All right? It's more detailed than I want you to memorize. Okay, so all you just have to, what you should understand is this is different from a simple activation. All right, fructose metabolism, my personal favorite, because fructose, okay, if you go out and go to your tea shop, okay, they're going to add a bunch of fructose in your drink. Okay, so you're going to be drinking a lot of fructose. Okay. Fructose, like I mentioned previously, it has 2 mechanisms for degradation. The first one is through hexokinase, like glucose. Okay, so if you do hexokinase, you get F6P. F6P continues glycolysis. Fructose has an alternative pathway which you can use a fructokinase. And this fructokinase will generate fructose 1-phosphate. And then this fructose 1-phosphate can directly be sort of converted into glycerolaldehyde, not glycerolaldehyde 3-phosphate, just glycerolaldehyde, and dihydroxyacetone phosphate, DHAP, okay? And this is an aldolase. What you should notice is that using this pathway, you bypasses the regulation of F16BPase and the phosphofructokinase, okay? You bypass the most important step for glycolysis, okay? So basically what this means is that this fructose is going to be utilized for energy or lipids generation. 有听懂我意思吗？就是说，这个很重要，因为我们不是说 glycolysis 最重要的一个 regulation step 是把 F6P 变成 F16BP 吗？ Okay, 那是 glycolysis 最重要的一个 regulation step。那问题是说 fructose 的 degradation mechanism 里面呢，其中有一步就是如果你选择用 fructokinase， 然后你去做出这个 fructose 1-phosphate 那个 F1P， 那这个时候你可以直接用一个 aldolase 直接把它切出来，切成格索aldehyd跟 DHAP。那这一步就等于完全跳过了 F6P 到 F16BP 的那个 regulation step。OK， 那 what happens then is that 这个会告诉，其实这会告诉你一件事情哦，就是说你的这个 sugar 基本上就是要被 either 烧掉，不然是变成 something else。OK， 就是你不受调控。OK， 你这个的 carbon flux 等于不受调控。Okay, so once you get glyceroldehyde and then DHAP, DHAP obviously can continue to go glycolysis into glyceroldehyde 3-phosphate through our triose phosphate isomerase. Okay, this is glycolysis. Okay, now turns out you get this glyceroldehyde. Okay, this glyceroldehyde, and glyceroldehyde can also take ATP. And what happens is that you can convert glyceroldehyde to glyceroldehyde 3-phosphate. So you're probably thinking, hey, that's fine because even though you bypass the most important step for glycolysis regulation, but in the end you still get glyceroldehyde 3-phosphate, okay, which is okay. But what you should understand is that once you bypass the regulation and you have a bunch of DHAP, either it goes back to do gluconeogenesis or it's going to do this. Okay, turns out dihydroxyacetone phosphate can get reduced into glycerol 3-phosphate. Glycerol 3-phosphate is the direct precursor to making your triacylglycerol. Okay, because glycerol 3-phosphate is like the the head for making lipids. Okay, so this is people suspect this is one of the causes for obesity, for excessive fructose consumption. So obviously you hear in the news saying that, oh, you shouldn't eat too much fructose because it's bad for you and it causes like cardiovascular diseases if you expose it to a long term. Okay, one of the reasons people think is probably due to obesity associated with fructose. Okay, right? So if you want to drink something sweet, at least you get sucrose or something. Don't get just all fructose. Okay, glycerol. So let's talk about glycerol. Okay, glycerol, 甘油. You will get glycerol from different places. Some people, glycerol is edible. It's not like toxic. It's edible. It's like nasty. It's like greasy kind of, right? But nobody really eat glycerol. What happens is that you get glycerol from lipid metabolism. Okay, so when you sort of degrade your lipid, you can get some glycerol. And this glycerol, the way to utilize glycerol is that you activate C3, okay, into glycerol 3-phosphate. And then glycerol 3-phosphate can do an oxidation to DHAP. So basically the reverse of this reaction. Okay? So that's how you use glycerol. All right? So we talked about glucose, fructose, galactose, and mannose, the other sugars. Talked about glycerol. Okay? All right, so those are the monomers. Now I'm going to spend a little bit of time talking about polysaccharide degradation. Okay, polysaccharides. because polysaccharides, there are different types of polysaccharides, and I want to sort of have you appreciate the different types. Now, on day 1 of our class, we talked about like amylase, the enzyme that degrades amylose. What it does is it's going to use water, and it's going to do a hydrolysis, and you directly cut this bond. And then you generate 2 monomers, this and that. Okay? Sounds good. However, this is thermodynamically favorable, so you think maybe I can sort of conserve this energy. So if I want to conserve this energy, I can do this thing called phosphorylasis, which I use PI, inorganic phosphate, and use PI to do this breakage, okay, this breaking reaction, lysis reaction. So it's called phosphorylasis. And then you get sugar 1-phosphate, which is glucose 1-phosphate, and then you get like glucose, okay? So you get phosphate. Now this guy has energy. If for degradation, you can directly convert this guy basically to G6P, okay, without adding ATP. So 2 types of reaction exist for liberating a monosaccharide unit. There is the hydrolysis, typically used for digestion of dietary carbohydrates like starch, okay? The reason for that is free sugars are easier to transport into the cell, okay, I forgot to into the cell, whereas sugar phosphate are harder to leave the cell, okay? What that means is you can't just all of a sudden have a transporter for sugar phosphate. Okay, because if you have a transporter for sugar phosphate, maybe sugar phosphate can come in, it can come out. We don't want that. We don't want that. Okay, so what we want is to have transport of sugar and not sugar phosphate. Hydrolysis typically occurs for dietary carbohydrates like starch, that kind of stuff. Phosphorylasis is usually used for degrading intracellular carbon storage like glycogen or that kind of stuff, okay? And that is beneficial, okay, this is beneficial for utilizing intracellular carbohydrates because phosphorylated sugar can directly feed into glycolysis like this, okay? Now, let me show you the difference between the 2. This guy is your cell. Okay, this guy is your cell. Your cell, okay? Let's say today you eat bread. Okay? You eat bread. Bread is like polysaccharide, amylose. Thank you, amylose. Amylose is like there. Cell, okay? This is amylose, cannot directly transport. This thing doesn't happen, right? Why? Too big, okay? Too big. This thing is too big. So this direct transport does not happen, okay? So you have 2 choices. Well, let's talk about hypothetical, okay? So you can do hydrolysis, okay? And you get this plus like that, okay? This guy? Okay, transport, simple transport. If you do, okay, here I say if all, if 不是 abbreviation, 是 if, okay? Hypothetically, if, okay? You do phosphorylasis, then what you should get is a sugar phosphate plus the remaining sugars. Okay? This guy? If this can come in, then pretty much there's a high possibility it can come out. Okay? So we don't want that. We don't want any sugar phosphate to leave the cell. Okay? Therefore, we do not have a transporter. This does not happen. Okay? Therefore, for most dietary carb, we're going to use hydrolysis. Okay? Now, if it's internal storage, I hope you guys understand that glycogen does not leave the cell. Okay? Glycogen does not leave the cell. Internal storage, this guy is glycogen. Okay? And you have two choices still. Hydrolysis or phosphorylasis. Okay, like that. In order to subsequently use these things, basically we need to do glycolysis. Right, glycolysis. Let's say the first intermediate in glycolysis was G6P. This can directly convert, but this guy convert here, you need to consume ATP. Okay, so for conserving energy, intracellular storage, so internal storage, will use phosphorylasis and not hydrolysis, typically. Okay, but sometimes it's different. Okay, but typically. Makes sense. Okay, this makes sense. Okay, so so we're good with this difference between hydrolysis and phosphorylasis. Now, let's briefly take a look at amylopectin and glycogen. Remember what amylopectin and glycogens are? Glycogens are the sugar, sort of the short-term energy storage for us, okay, and for bacteria as well. Okay, so it's sugar polymer, which can sort of just stay there, and then when you need it, you degrade the sugar. Okay, it's a short-term energy storage. Long-term energy storage for you is, remember a couple of things. First of all, remember on your, I think what was it, like final exam or something like that, last semester, there was a question that says, you know, why do you think glycogen have a lot of branching? Right, that is because you can have more access to liberating the sugars. Okay. Now, in order to degrade amylopectin and glycogen, they're very similar. So what these things are, they're just, you know, highly branched sugar polymer, sort of. Okay, highly branched. Now, amylopectin is branching. And anytime when you have a branching, you have an alpha 1,6 bond between the two monomers. Okay, hopefully you still remember that, because typically if you have a linear chain, right, the linear is alpha 1,4. Okay, so amylose, okay, amylose is straight chain alpha 1,4 glucose. So glucose, alpha 1,4 linkage, and just linear chain. That's amylose. So alpha amylase, okay, amylase then degrades amylose. Simple. Glycogen degradation, glycogen phosphorylase will catalyze the phosphorylasis of glycogen to glucose 1-phosphate. Okay, so like that. So if you do amylase, okay, amylase, you can degrade from 1,4, like 1,4, kind of 1,4, like that. I don't have glycogen phosphorylase here, but once you get down to the branching, you no longer can use alpha amylase to do this, because amylase only do alpha 1,4, okay? So you need to have a different enzyme. This enzyme is then called alpha 1,6 glucosidase. So it's glucosidase, meaning it degrades glycosidic bonds, and it's specific for alpha 1,6. Okay, so therefore the name is very straightforward. So once you do that, you can cut these things off. Cut them off, okay? And then you can continue to degrade the linear chain until you reach the next branching. In glycogen, okay, glycogen obviously you can degrade linear using glycogen phosphorylase. Okay, so the linear part is also degraded through phosphorylasis for glycogen, but for amylose and amylopectin, it's through amylase. 如果你今天要 degrade amylose, okay, 淀粉里面两个最大的东西 amylose and amylopectin. Okay, so if you want to degrade amylose, amylopectin, you're going to use hydrolysis, okay, and then you're going to follow sort of this thing. If you're degrading glycogen, glycogen is usually inside of the cell, you're going to use glycogen phosphorylase. For glycogen debranching, okay, so this mechanism, this is for amylopectin. If you want to degrade branching or get rid of branching for glycogen, you need to have a special mechanism, and this special mechanism is called glycogen glucan transferase. It's shown in this little picture here. Okay, so this you guys understand, eh? So for amylopectin, it's very simple, where you do linear degradation until you reach the branching, okay, the branching. Then you use a different enzyme called alpha-1,6 glucosidase, and then get rid of the branch and then continue. For glycogen, it's different. It's not through alpha-1,4 degradation. What it's going to do is, after you do phosphorylation to a short sugar polymer here, what you're going to do is you're going to do a transferase reaction where you transfer these three from one branch to the other branch, like that. Okay, so then once you transfer that, you can get rid of the branching here. Okay, so it's not through 1,4,1,4, not like amylopectin. Okay, all right. So do you understand? Okay, so you see, initially you started with 3 here, you ended up with 6 here, excluding the branch. Okay? You have to use that. That's why I was saying, like, it's a transferase reaction. Okay, this is a transferase. So you transfer these 3 from one branch to this branch, and then you can use hydrolysis to get rid of this guy. So this is how you degrade amylopectin and glycogen. Okay, sounds good? Now, glycogen synthesis. So we talked about glycogen degradation. So now we're going to talk about glycogen synthesis. So glycogen synthesis is going to start from glucose 6-phosphate. Okay, it's going to always start from glucose 6-phosphate. Now, glucose 6-phosphate, what you have to do is turn glucose 6-phosphate to glucose 1-phosphate. Okay, and then after you get glucose 1-phosphate, what you're going to do is you're going to hook up UMP. This guy's UMP onto this phosphate, and then you generate this UDP glucose. You saw this on the slide where we had galactose degradation. Okay, and then once you get UDP galactose, then you can take a glycogen primer, so basically a terminal end of a glycogen. Okay, this is the terminal end, and then you're going to sort of hook up from here, this part, onto here. Okay, and then you elongate the chain. Okay, make sense? Because if you kick out UDP, UDP is a very good leaving group because the phosphate can delocalize its electrons. So kicking out UDP is favorable, and you can then elongate the chain. Okay, so UDP glucose is metabolically activated form of glucose or for glycogen synthesis. Okay. Here's a quick question for you. If I ask you a question of which step would be the regulated step in glycogen synthesis, and what do you think? Probably like this step. Okay? Right, because glucose 1-phosphate can sort of stay in glucose 1-phosphate. Glucose 6-phosphate can be used for other stuff. But if you activate this guy using UTP into UDP glucose, it's going to be used for something. And especially if you look at the energy, here is quite large, minus 19. So probably this step is going to be somewhat regulated. What I should point out is that, what do you notice about this particular reaction? This particular reaction, going from glucose 1-phosphate plus UTP to UDP glucose, okay, the energy here is around 0. But it's when you have pyrophosphate degrade to 2 phosphates that gives you minus 19. Okay, so a lot of times when you do hydrolysis, when you use, for example, ATP to AMP, okay, the two phosphates, when you get it, it's one pyrophosphate. Pyrophosphate is the two phosphates are still together, okay, and subsequently the pyrophosphate can hydrolyze to two phosphates. That is the driving force. So in terms of this, you probably regulate this step. Yeah, because that one is probably. You should be able to answer this question: why is branching important for glycogen? For the access of this, right? So you can quickly access the different glucose. Why is branching important for glycogen? Okay. So turns out if you want to make glycogen, glycogen synthesis also requires this thing called glycogen synthase. Right here, glycogen synthase. Glycogen synthase for the polymerization, okay, so for polymerization you use synthase. But if you need to do branching, because remember we say that glycogen has a lot of branching because it wants to be able to access from many different points. Now this branching is introduced by this transglycosylase, okay, transglycosylase. The way it works is that you first isolate this glycogen, and then this is the outside, okay, this is outside, this is the glycogen core. And what you can do is use an enzyme that will connect this particular sugar with this particular sugar through a alpha-1,6 bonding. Okay, so you transfer this particular glucosidic bond to this particular glucosidic bond. Okay, so it's a transglycosylase. This cascade, I think it's pretty straightforward to you guys because we spent so much time on the first part of our class today talking about regulation. So the liver cell, hepatocytes are liver cell, that's liver cell, glucagon, that's what you saw like an hour ago, okay? Where you have glucagon coming in, it's going to activate adenylyl cyclase, so it's going to turn ATP into CAMP. Okay, so this part you saw already. Okay, you saw this already. Glucagon coming in, activate CAMP, AMP activate protein kinase. Protein kinase, it's going to phosphorylate this other kinase to make this guy active. This guy is going to affect this guy, and then this guy is going to do a lot of catalysis. And what that's going to do is degrade glycogen. Okay, now, what this picture wants to show you is the significance of signaling cascade. Okay, so just by having one molecule, one signaling molecule from the outside, okay, this one particular signaling molecule can turn on an enzyme. This enzyme is turned on. This enzyme is going to catalyze a bunch of reactions. Let's say for every glucagon molecule you bind to the receptor, and it's going to trigger this particular enzyme for converting ATP to AMP, cyclic AMP. And for every one of this, you get, let's say, 20 of cyclic AMP. 20 cyclic AMP is going to then activate this particular protein and give you 10, and then this one can give you 100, and then 100 to here can give you 1,000. By the end, you get like 10,000. So this is an amplification effect, okay? Because every time when a molecule binds and activates an enzyme, this enzyme is starting to do catalysis. It's starting to do catalysis, okay? And once it's doing catalysis, it can create more than one molecule. Okay, so this is not a linear translation of message. This is the amplification of the message. Okay, this is why you have so many levels of signal transduction. Okay, you can actually amplify this message, right? Because every time you turn on an enzyme, it's going to make a bunch of molecules, and then these bunch of molecules will affect the next level of enzyme, and that enzyme is going to make a bunch of molecules. Okay, does that make sense? Right, now, so in the liver cell, here we have, this is what happens in the liver cell. Now, want to mention something else. This is myocytes in muscle cells, okay, or blood cells, primarily muscle. In the muscle, you have this particular thing called, I usually just call it adrenaline. Okay, adrenaline 就是那个肾上腺素是不是 Okay, adrenaline. So what is this is, you see in the movie all the time when like people have high adrenaline, they get like this, they get very like, not excited, but you have a harder, faster heart rate, okay? You basically prepare yourself for emergency, for a state of you have to be able to quickly respond to different kinds of situation, right? That's adrenaline rush. So if you go on a roller coaster, that's what you get, okay? Because all of a sudden your body is getting accelerated, so then your cell, your brain, okay, it's signaling to the rest of your body saying you might be in danger. 你听懂我意思吗？坐云霄飞车的时候有没有？你不是会那个心跳会加速，心跳加速就出现这个反应 ，OK？ 因为你的人会忽然被甩到一边或甩到另外一边，然后这个时候你的大脑会跟你全身说something's wrong 哦 ，OK？ 要 prepare for something, okay? Now, the way it works is pretty much identical to what happened in the liver cell, okay? So it's pretty much the identical set of things that you're going to do. And basically what it accomplishes at the end is to very quickly, very fast, okay, very quickly degrade a bunch of glycogen into glucose phosphates. And then this glucose phosphates can then be used for glycolysis for muscle contraction. Okay? All right, so in muscle, this is what we want to do. Because in a state of emergency, your brain will send out these signals, okay, adrenaline, they're sending out these signals that's going to tell your entire body to prepare for war. Okay, prepare. Right? And in order to prepare your body for emergency, okay, like for emergency, you need to have a lot of energy. Okay, that's for muscle contraction. And in order to get energy, you need glycolysis. Okay? So this rapid mobilization of muscle glycogen triggered by this adrenaline is one of the several components of the what we call fight or flight response. Okay? So either fight or you run away. Okay? So either of these require a high amount of energy mobilization. Okay, so you have to very quickly make response. Okay, so this is how it works. Is that clear? Okay, your brain sends out the signal, and then very quickly you have a fast response. And how do you get the fast response? Because you cannot rely on your brain to make 10,000 adrenaline in like 5 seconds, okay? So you have the signal transduction, and every step in that 5 seconds, you can amplify this signal. So that's how you get a quick response. Okay, ma? All right, so half an hour, just enough time for pentose phosphate. So we talked about sugar degradations, and we talked about some of the interesting regulations for sugar, okay? Now, the next thing that we're going to talk about, most people will not like this because this is a very, in a way, confusing pathway, but it's a pathway that is super important, okay? So there's this thing called pentose phosphate pathway, and pentose phosphate pathway is sort of the key pathway for generating NADPH, okay? And it's also used to generate ribose 5-phosphate. Okay, ribose phosphate is used for making DNA and RNA. Okay, so obviously this pathway is very important. Okay, now NADPH is important for what? NADPH is important for making biosynthesis, right? Biosynthesis of like lipids, amino acids, whatever. So anything have to do with biosynthesis, you're going to use NADPH. Okay. So NADPH is very important, okay? So they're going to use it for fatty acid, cholesterol, neurotransmitters, nucleotides. It's also going to be used for some detoxification reactions that are very important, okay? So NADPH is important. And this particular pathway is sort of the major pathway for us to generate NADPH, okay? There are several, but this pathway is one of the major ones, okay? Now, also very important for this particular pathway is that it generates different sugar phosphates, okay? Different carbon length sugar phosphates, okay? PPP. So what happens is in glycolysis, the first intermediate you have is glucose 6-phosphate, okay? And glucose 6-phosphate will first go through a phase. Okay, we can divide the pentose phosphate pathway into two phases. The first phase is the oxidative phase. The second phase is the carbon rearrangement phase. Okay, the first phase is going to do a bunch of reactions, and essentially it will turn glucose 6-phosphate into ribulose 5-phosphate. Okay, what happened here? You lost one carbon. Okay, it's not shown on this picture, but you lost one carbon. But in addition, okay, what you can notice is that 5-phosphate probably indicates that it has 5 carbons. Yeah, probably. Okay, so going from glucose 6-phosphate to R5P, ribulose 5-phosphate, you lost one CO2. So the energy from that one CO2 is turned into NADPH. Okay, from that one carbon. Okay, because glucose 6-phosphate and ribulose 5-phosphate oxidation state is sort of the same. Okay, because sugar is the same sort of oxidation state, right? Because it's always every carbon is hydroxyl group except one carbon is a keto group. That's what makes it a sugar. Okay. Now, ribulose 5-phosphate can do a couple isomerization reaction. It can become ribose 5-phosphate or xylulose 5-phosphate, and then you're going to do a series of recombination, and then at the end you can end up with fructose 6-phosphate and glyceraldehyde 3-phosphate. Okay, we're going to talk about this in detail. Now, this is the oxidative branch of the pentose phosphate pathway. Okay, and basically, like I said, in the oxidative branch, one carbon of G6P is oxidized completely to CO2, and that gives you 2 NADPH. Okay, so G6P becomes R5P. So this part is sort of linear. Okay, so it's a linear sequence. You start with glucose 6-phosphate, and you're going to oxidize that. Okay, because you produce NADPH. Okay, so you oxidize. So in this direction, it's an oxidation, and you make this 6-phosphoglucanolactone. Okay, so it's a lactone. Did I tell you the difference between lactone and lactam? Lactone. Lactam. Okay, and these could be N carbons inside. Okay, it depends on how many carbons it's going to be called a different lactone. Okay. So here you can see how there's a ring structure right here. Okay, ring structure, and this guy is a lactone because here it's carbonyl and oxygen here, so it's an acid. And then what you can do is you can do a lactonase, which you cleave open this particular lactone structure, such that you end up with 6-phosphogluconate. Okay, so this is an acid. Okay, so essentially what you did was you take G6P, which is a sugar, right? Initially here was basically an aldehyde, and then you turn it into a sugar acid. Okay, so now this is an acid. And what you're going to do is you're going to do a decarboxylation of this guy. Okay, and you do a dehydrogenase reaction, okay, dehydrogenation, where you're going to sort of oxidize, so here's 1, 2, 3, 4, 5, right? So you're going to oxidize this guy from alcohol to a ketone, right? Okay, so this carbon and this carbon is the same. Now, if you oxidize this alcohol into a ketone, what is this acid? It's a beta keto acid, right? Acid, alpha carbon, beta carbon, okay? So if this carbon now is a keto group, then this becomes a beta keto acid. And remember I said beta keto acid decarboxylates spontaneously, okay? So then you are left with this guy, ribulose 5-phosphate, okay? So the oxidative branch for the PPP, I call this guy PPP because it's pentose phosphate pathway, PPP. The oxidative branch for PPP, it's pretty simple. It's 2 sequential oxidations pretty much, okay? You oxidize the aldehyde, okay? Initially you oxidize the aldehyde and it becomes an acid. And then what you do is you oxidize C3 from an alcohol to a ketone, and because now it becomes a beta keto acid, the acid will decarboxylate, become CO2. And ribulose 5-phosphate, this is a ketone, so this guy is a ketose or aldose? 它是 ketose, okay? So 它是 ketose. So this guy is a ketose, and if you do isomerase reaction, you can turn it into ketose to aldose. So ribose 5-phosphate is aldose, okay? 就是你看哦，就是我们到这一步的时候 OK 嘛，对不对？就是你把 aldehyde 变成 lactone 水解出来变成 acid， 然后这边是 acid， 然后呢，就是这个 structure 跟这个 structure 是一模一样的啦 ，OK。And then what happens 是我们就要做一个 oxidation， 所以我 oxidize 是这个，原本是 OH 嘛，那我把这个 OH 变成 C double bond O， 变成一个 carbonyl， 然后它就变成 beta carbon， 对不对？它本来就是 beta carbon， 它不是变成 beta carbon， 所以上面是 acid， 然后这个是 alpha， 然后这个是 beta。那如果这边变成 ketone 的时候，它就是一个 beta keto 的 acid，right？ 那 beta keto acid 可以 spontaneous 的被 decarboxylate，okay? And then that's how you get 这个 ribulose 5-phosphate。那这个是 ketose， 我们可以再把它变成 aldose。那这个是 reversible，okay? Pay attention to the reversibility shown here, okay? Like this is one way, this is one way, this is reversible, this guy is reversible, okay? All right. So the oxidative branch, I think it's pretty straightforward, okay? You take G6P, you get ribulose 5-phosphate or ribose 5-phosphate, okay? So G6P to R5P, okay? G6P, R5P. In the middle, you kick out 1 carbon dioxide and 2 NADPH, okay? Now, this part, it gets a little complicated, okay? This part is where it gets a little complicated. This is the non-oxidative branch of PPP, okay? But what I usually call it, it's carbon rearrangement. Okay, so we're going to be able to get all sorts of different sugar phosphates. Okay, sugar phosphates. All right. So in the oxidative branch, nothing happens. Okay, seriously, you don't get any oxidation. You don't consume ATP. You don't consume anything. All you're doing is number rearrangement. Like 5 plus 5 gives you 3 plus 7. 3 plus 7 gives you like 6 plus 4, things like that. Okay, so in this phase, nothing happens except carbon rearrangement. Okay, you have to understand that. All right, and the enzymes used in this pathway, this part of the pathway, there are a couple. There's the epimerase, there's the transketolase, and there's the transaldolase. Okay, we'll talk about those in just a minute. Okay, but for my purpose, I'm more interested in you understanding how do we get different things, these blah blah phosphates, okay? Now, at the end of the oxidative phase, we are left with ribose 5-phosphate, okay? And obviously, ribose 5-phosphate can be used to make DNA, RNA, okay? Deoxyribo blah blah, okay? So, ribose 5-phosphate, that is used for making DNA, RNA, okay? And some of these can go to central metabolism. Erythrose 4-phosphate is used to make aromatic amino acids. Okay, so all of these have purposes. Now, so at the end of the oxidative phase, you get ribose 5-phosphate R5P. So R5P can do epimerase to make xylulose 5-phosphate. So this is reversible. Okay, and then what you can do then is do a transketolase reaction. So 5 plus 5, you get 7 plus 3. Okay, shown over there. Okay, so cetoheptulose 7-phosphate and glyceraldehyde 3-phosphate. Okay, but 7 and 3, 7 is not used for anything. Okay, it's not really used for much. So what that's going to do is you're going to couple that with a transaldolase, which converts 7 plus 3 gives you 6 plus 4. Okay, so aldolase move 1 carbon and transketolase move 2 carbons. Make sense? Because 5 becomes 7, 5 becomes 3. And here's 3 becomes 4, 7 becomes 6. So transketolase move 2 carbon, transaldolase move 1 carbon. Okay? If I want to completely convert my ribose 5-phosphate back to central metabolism, which is C6, what you can imagine is I want to get C5 back to C6. What do? You take common denominator, which is 6 C5, okay, can give you 5 C6. 合理吧? Okay? So using this, you can think of all sorts of different carbon rearrangement. And I'll show you why this is important later today. Okay? Epimerase, basically, you just swap this guy to that side. Epimerase, basically, you swap the stoichiometry. So obviously, you're going to do bond breaking and bond reforming. Okay? 这个懂吗? Okay? 因为你没那个 stereo center 不能就 randomly 就是这样飞哦。Okay? 所以你会把这个拔掉，然后插上去。Okay? Transketolase is kind of interesting because you're going to move 2 carbons, okay, at a time. And basically what happens is you're going to put this carbonyl and you're going to basically put it right here. The initial aldehyde becomes a hydroxyl group, okay? And then this becomes an aldehyde. So you sort of, you move the keto part, this entire part, okay? Right? And this you can do it for xylulose 5-phosphate plus urethros 4-phosphate can give you glyceraldehyde 3-phosphate and fructose 6-phosphate. Or you can use the keto donor is from xylulose 5-phosphate, same, but then your aldose is ribose 5-phosphate, and then you get 3 plus 7 or 3 plus 6. Okay. Basically, what happens is you always have a ketose and an aldose. And ketose minus 2. Aldose plus 2. Transketolase reaction reacts ketose and aldose. Ketose will be 2 carbon shorter, aldose will be 2 carbon longer. Okay? That's transketolase. And the way it works is like this. You are going to require this TPP cofactor, okay, to transfer its 2 carbon unit as an activated aldehyde. So again, TPP can become this elite form TPP carbanion. And this minus charge, this nucleophile, will attack the ketone. And you're going to form this tetrahedral intermediate, which then you can pop this particular electrons down here, forming another ketone here. You cut this bond. Okay, so pretty much this 2 carbon here. So you see how this is the terminal 2 carbon, right? So the terminal 2 carbon, this is the keto. That's the hydroxyl group. These two get attached onto TPP like this. So your original ketose now is released as an aldose. Okay? And the aldose substrate comes in. Okay, so this is now the aldose come in. And you're going to do a nucleophilic attack on the aldose. Okay, and you reform this tetrahedral intermediate, which then you can then kick out this guy and you reform the ketone, and then you're left with the ketose product. This is the terminal 2 carbon, right? This is, for example, this is, let's say, fructose or whatever, fructose 6-phosphate. Here's the ketone, right? And then here is the terminal C1, C2, right? And this TPP attacks C2. You generate the tetrahedral intermediate, and then you can cleave this particular bond because you can generate an aldehyde from the original ketose. You generate an aldose product, okay? And then the 2 carbon of the ketose is now on the TPP. Then you take your aldose substrate come in, and those 2 carbons basically get attached to the aldehyde, and originally you have an aldehyde, you ended up with an alcohol. Okay, so that's how you always, for transketolase, it's ketose. If, let's say, you start with 5 carbon, at the end, you leave with 3 carbons, okay? And the aldose gets plus 2. Okay, so ketose minus 2, aldose plus 2. Okay, and the reactant is always 1 ketose, 1 aldose. 可以吗？就是 transketolase 的 reaction， 你 always 是一个 ketose 加一个 aldose， 然后出来一个 ketose 加一个 aldose。OK， 那你放进去的 ketose 呢，出来的时候会减2个碳，然后你放进去的 aldose 呢， 出来了以后会变成多2个碳。Okay, so this is transketolase. Okay, transaldolase. Transaldolase it's kind of crazy because you're going to use that shift base thing again. Okay, remember we talked about shift base when we first talked about aldolase. Remember that? When we talked about aldolase in glycolysis, we talked about shift base. Okay, so shift base is where you take a lysine amino group, replace it with this particular keto, okay, and then you generate this shift base. Okay. If you want to look at how this thing happened, go back to reaction 4 of glycolysis. You will find out. Right? Now, what you can have is a general base that will deprotonate this particular proton, and then this particular proton sort of come here, and you can sort of leave this 3 carbon, you leave it, and you end up with this urethral 4-phosphate. Okay, so you sort of kick out 3 carbons. So 3 carbon here, 4 carbon here. Okay, initially this is 7 carbons. Okay, the 4 carbon leave as a product aldose. The substrate, okay, the substrate here is a glyceraldehyde 3-phosphate. Okay, it's an aldose. Right, the substrate aldose comes in, and it's going to do a nucleophilic attack on the aldose carbonyl. And then you will then generate this new bond between the 3 carbons on your enzyme, and you generate that bond between the 3 carbon on your enzyme with the aldose, and you form this bond. You do reverse shift base formation, and you're left with a ketose. Okay, so you take the 7 carbon becomes a 4 carbon, and the 3 carbon here becomes a 6 carbon. So take what I said earlier back. It's a 3 carbon transfer, not 1 carbon transfer. But if you look, it's 7 becomes 6, and then 3 become 4. Okay? But it's really a 3 carbon transfer. You know, you can compare the CC bond cleavage in transketolase and transaldolase reaction. But again, you know, this you can take a look when you have a chance to review this back home. You look at these 2 mechanisms. The transketolase mechanism and transaldolase mechanism, what you will always see is that you will get this C double bond N thing right here. In the transaldolase, it's a shift base. Transketolase is TPP. Okay, you always have this electron sink that draws away the electrons. Okay, so their catalysis, although they use different cofactors and different mechanisms, but in reality, their mechanism is very, very similar. Okay. PPP is actually very important because you can think of several modes of operation. Okay, several modes of operation. What does that mean? That means that what you can do is, let's say I want to produce only NADPH for biosynthesis, and I do not want ribulose or ribose 5-phosphate. Can I do that? Yes, you can. After the oxidative part, okay, after the oxidative part, you generate RU5P, okay, ribulose 5-phosphate. Okay, so you generate NADPH. Now, I don't want to use RU5P. I want to recycle RU5P back to G6P. Can I do that? Yes, because you can do a series of, 5 plus 5, 7 plus 3, 4 plus 5, etc. So you don't do anything with the carbon. All you do is recycle G6P back up. Okay, so you can think of one extreme is I only make NADPH, okay, and then all the other carbons I recycle back to make C6, so that I can continue burning my carbon to generate NADPH. That's one extreme operation. Okay. Now, another extreme operation is where, okay, today I maybe want to use both my NADPH and my carbons, for example. Okay, so here what I can do is I do my PPP and then I generate it to RU5P. RU5P can then go to F6P, G3P, and then they continue glycolysis, and then they all gets converted to CO2. Okay, so this is also sort of doing a energy generation. Okay, So I'm going to oxidize all of my carbons. I would do that. Okay? Or what you can then also do is, okay, what you can also do is, let's say today I'm interested in making DNA. Okay? DNA, I need to have R5P and an ADPH. Okay? So then you can stop at R5P and then take R5P to DNA using an ADH. Okay? I can also do that. Right? So basically, if you look at PPP, what PPP allows you to do is flexibility. Okay? You can do a lot of different things using the pentose phosphate pathway. You can use it to only generate NADPH, or you can use it to generate NADPH and the ribose, and you use that to make DNA. Or you can just use that and couple it with glycolysis to then convert all of your carbons into CO2. You can do that as well. Okay? So PPP is a very important pathway primarily for two things. Okay? So in summary, two things: NADPH generation and diverse sugar phosphate generation. For example, R5P for DNA. Okay? Right? So this is probably the most confusing lecture you'll ever have.